And I'll also be introducing our next speaker, Julia Schaepe. Julia comes to us from Stanford University, where she's a student in the lab of William Greenleaf. And Julia is working on a very important research area in the context of gene expression. Uh, she and her colleagues in the Greenleaf lab have developed a really elegant approach to understand in very intricate molecular details how enhancers and activation domains and transcription factors coordinate together to coordinate gene expression. And this is a really important question. Many of you are probably aware that variation in enhancers, variation in activation domains are a very important link between genetic variation and differences in gene expression, and probably ultimately differences in things like health and disease. And so, what I think is exciting about Julia's work is it's really moving us from this sort of binary models on off to a really quantitative and mechanistic understanding of how these genomic features coordinate gene expression. So, please give a warm welcome to Julia.
Thank you so much for such a kind and thoughtful introduction, and thank you for the invitation to come talk. As you could tell from the introduction, not in the single-cell proteomics space, so I've really enjoyed learning more about this field. And in this talk, I'll transition from the last talk of more cell heterogeneity to thinking about molecular heterogeneity, and specifically thinking about how transcription factors interact with DNA to regulate gene expression, and how hopefully you can take away from this some of the methods we use to be able to get really high resolution and quantitative measurements that allow us to actually use really simple physical models to understand what's going on. And the work I'll be talking about today is the first project led with Ben Dowdy and Michaela Hinks through a really fruitful collaboration with the Bintu Lab. And the second project I'll talk about is co-led with Emil Markland, who's a professor at Stockholm University. And so, as many of you know, transcription factors, these proteins, are very important in regulating gene expression. But there's many molecular details that are not yet quantitatively understood in how they interact with DNA and form open chromatin within cells. Historically, we've studied these with bulk genomics approaches like RNA-seq, ATAC-seq, or ChIP-seq. I don't think I need to convince you all why moving to single cell or single molecule is useful and informative. But specifically, we're interested in if we can see underneath one of these ATAC-seq peaks to understand the individual and heterogeneous states that make up one of these peaks. To illustrate this, I'll talk about something that's been seen in the field of genomics since the 90s, that as you linearly increase your motif copy numbers, so these little blue boxes denote transcription factor motifs on DNA, as you linearly increase the number of those, you get nonlinear increases in gene expression. And over the years, there's been many hypotheses for why we observe this, whether it comes from cooperativity in binding or cooperativity in activation once you're bound. But because we don't have these molecular measurements, it's been really hard to say which one it is. And so we think what you need is to actually measure the transcription factor occupancy that links your motif number to the ultimate gene expression. So how are we going to do this? We built a very simple synthetic system in which we have a minimal promoter driving expression of the fluorescent reporter citrine. And then upstream, we installed a library of enhancers that have variable motif numbers for the synthetic transcription factor RTDR-VP48, which is DOX-inducible and drives very strong gene expression. We then integrate this at AAVS1 and K562 cells, so they're within the genome. And we can confirm that as you increase this motif number, you do indeed get these nonlinear increases in gene expression as measured by flow cytometry. So in the system, how are we going to measure transcription factor occupancy? To do this, we turn to single molecule footprinting. So this was heavily inspired by the work of Arno Krebs and others. And so how it works is we take our cells, we then incubate them with the GPC methyltransferase, which adds these methylation groups to accessible sites along DNA, giving us a really high resolution and contiguous picture of accessibility. We can then convert this accessibility with an enzymatic version of bisulfite sequencing, and then we just read out these molecules on a standard Illumina sequencer. What's nice about this is that different molecular states will have different and interpretable molecular patterns, such as nucleosomes having larger protection widths or even seeing transcriptional machinery at the promoter. And so to illustrate this point, here are 4 actual molecules from our data set. Up is accessible and down is protected. You might be able to label what's going on by eye, but we built a probabilistic model based on prior assumptions to be able to infer these states. And so for every single molecule, you can count how many transcription factors are bound at the enhancer and whether the promoter is in an active or nucleosomal state. So basically, is the promoter bound by a nucleosome or is it open and accessible? We don't just have 4 molecules. We measure thousands for every single construct that we put in the system. And so here we have 8 motifs in this molecule, and we have each row here is an individual molecule that we've measured, and every column is a site that we get methylation information at, where white is accessible and black is protected. So these stripes of black underneath these motifs represent TF occupancy. And so we can see this now across the library with different motif numbers. And what you might appreciate is there's a really big jump between 4 and 6 motifs, so in that TF binding or those stripes that you see. The reason we're excited about this data is that now for the first time you can actually see what the distribution of TF occupancy is along molecules. So here, if we take molecules that all have 7 motifs, we see that some have all sites bound, some have none, and most have 3 to 4 transcription factors bound. And the reason we're really interested in this is because it gives us an opportunity to test these types of gene regulation models that have been long used by Locker-Bintu and others. And these partition function models are borrowed from statistical mechanics. And so we started with the very simple one that we could come up with, where there's an energy of interaction between the transcription factor and DNA, and an energy of interaction between the nucleosome and DNA. You then list out the microstates, so all of the possible ways your molecule could be, and then assign an energy to each one of those states. That energy then dictates the probability of observing it through the Boltzmann distribution. We then fit this across our data set, and you might be able to appreciate it does a really poor job. So clearly we're missing something. And we thought, okay, it's not just RTED-R, this DNA binding domain. It also has a separate part to this protein, the activation domain, VP48. That can interact with other cofactors that are known to destabilize nucleosomes, like chromatin remodelers like BAF or acetyltransferases. So we included a third parameter such that when the transcription factor is bound, it's able to recruit cofactors that then decrease the energy of interaction between nucleosomes and DNA. So it's destabilizing nucleosomes when the TF is there. Now with this one additional parameter, we can really nicely fit this bimodal distribution, and not just for molecules with 7 motifs, but across our whole library. So that's great. We tried other models, they don't explain it as well, but we still want to experimentally validate that this makes sense. So the first thing we did was we chopped off the activation domain. And what you can appreciate is that this leads to a big reduction in transcription factor binding. So that tells us that activation domains play a big role in how TFs bind within chromatin, which is really the first time we've been able to see that. And we can now explain it with this really simple competition 2-parameter model. We next wanted to know, who are the cofactors that are being recruited? So the first one we inhibited was the catalytic domain of P300, which is an acetyltransferase. And interestingly, that when we inhibit this catalytic domain of P300, we actually see a slight increase in TF occupancy. And so P300 is not the cofactor that helps stabilize TF binding in chromatin. Now, when we inhibited the ATPase domain of BAF, which is a known chromatin remodeler, we do see this drastic reduction in TF occupancy, consistent with these activation domains recruiting cofactors that destabilize nucleosomes, and this really drives your TF binding within chromatin. So far I've really talked about this side of the molecule, the TF binding, and now we can look at how that relates to the other side of the molecule, which are the promoters that we read out along the same molecule. And so we fit this nonlinearity, cooperativity, and TF occupancy quite well. And how does this occupancy then relate to the promoter state? Amazingly, and it didn't have to be this way, what we see is that each TF is basically contributing independently to promoter activity. So there's this really linear relationship in between TF occupancy and the promoter activity. This is a single molecule metric, but it actually has a linear relationship with gene expression for this system. That will not necessarily be true across different promoters, and as you all think a lot about, there is post-regulation that happens past transcription. But in this system, it is a linear relationship between those, and so TF occupancy directly relates to gene expression. And so we've been thinking about this as an additive activation model in which each TF is working independently to drive gene expression. The slope of this line we've been thinking about as potency. So per TF that is actually bound, how much transcription can it activate? And so you then would expect that within this transcription factor, if you tune its dosage, its potency should always stay the same. So the first thing we did was we tuned our transcription factor effective concentration by tuning DOX. So you can appreciate here that TF occupancy decreases, but when we look at its potency, or its ability to activate per TF that's bound, this stays the same. We were also curious if we could actually tune this potency by going back to this cofactor data. So if you remember, BAF inhibition tuned binding, but P300 did not. and so this is quantified on the right. When we look at potency, potency is decreased for both inhibitor conditions. So the ability to activate expression is dependent on both P300 and BAF. And so our models predict that since BAF inhibition hits both binding and potency, it should have a greater impact on gene expression. And indeed, that is what we measure by flow cytometry. So you might be like, this is totally a synthetic system, interesting, but how much of it actually applies to a human transcription factor? The system we turned to was type 1 interferon response, which is your cell's response to viral RNA infection. And so we installed these interferon-stimulated response elements upstream of the same reporter construct. And the way this system is thought to work is that pre-stimulation, some of these inactive subunits, IRF9, are actually bound, but you don't have active gene expression from those subunits. Post-interferon beta stimulation, you get phosphorylation of STAT1 and STAT2, you get the full active trimer that then translocates the nucleus and is able to stimulate gene expression. Interestingly, in our single molecule footprinting data, we can actually see these subunits, these narrow footprints, pre-stimulation, but we don't see active promoter states. And post-stimulation, we can actually see the widening of these footprints on individual molecules, consistent with being able to tell the difference between the inactive subunits and the fully active trimer. And when we look at these wide footprints and compare them to the promoter activity, this again holds with the independent activation model, and we can see potency being tuned by these cofactor inhibitions, albeit to different degrees than with VP48, since different cofactors are recruited by different activation domains. So to summarize this section, we built this reporter system to be able to connect measurements of TF occupancy and promoter state. We found that TFs compete with nucleosomes independently, but they get a really big boost by activation domains and the cofactors they can recruit. Once bound, they then work pretty independently to activate gene expression. And this cooperativity that we see across motif number is driven completely in this system by the cooperativity in TF binding. And so this is all focused on this one aspect of motif grammar, motif number. There's a lot of other aspects of motif grammar that we're interested in. And the one that I want to switch gears to talk about is how does sequence context outside of motifs actually tune transcription factor occupancy? To do this, we felt we needed to study this in vitro first, where it's just DNA and protein, and then move to the more complicated environment of cells. And so what we turn to is a platform that we use a lot in the lab. It's called HitsFlip. And so the way this works is that we design a library with tens of thousands of DNA sequences. Here we varied the motif, and we also varied the sequence context outside of the motif. You can then sequence this on a MiSeq, and then we actually image the chip while we flow fluorescently labeled protein on it. In this case, we chose KLF1. This is a human transcription factor that plays a really important role in erythropoiesis, and specifically the switch from fetal to adult hemoglobin. And so what this chip actually looks like, each cluster here is a cluster of DNA that we know the identity of. And so we can watch it light up over time, and from this we can extract association and dissociation constants between transcription factors in DNA, and we can also perform equilibrium experiments to extract affinities or the energies of these interactions across sequences. From that, we can construct position weight matrix, and so that is what the KLF1 motif looks like from this data. The way zinc fingers are thought of is that each individual finger, which this one has 3, interacts with 3 bases of DNA, and they tend to be very GC-rich motifs. And so this motif is very consistent with what is previously known for KLF1. What we were really interested in is what happens in these sequences where we're varying sequence outside of the motif. So here, there actually was really substantial effects, and that was pretty surprising to see. The first thing you can appreciate is that these 2 immediately flanking bases to the left of the motif, these 2 Gs, are quite important. It took a long time to think through this and digging into some old literature, but it turns out that this is likely due to direct contacts from an unconventional binding mode of this first zinc finger, meaning that those 2 should actually probably be considered part of the core motif, and the core motif I'm defining as something that is directly contacted when specifically bound. However, if we take those 2 out, there's still all of these more distal effects that can contribute over 2 KT or 10-fold effects on affinity. And so that's pretty substantial, and it really has this preference for more AT-rich sequence in the flanks. And so there's a lot more to dig into there, and I'd be happy to take questions, but a big question is, does any of this matter in cells? To do this, we turn to base-resolved deep learning models. So this is BPNet, and these are trained on chromatin immunoprecipitation data. And so from KLF1 data, you can train this to take in any DNA sequence and then predict what your chip profile is going to look like from that. And the reason these are so powerful is you can then do in silico computational experiments where you fix one base, whether that's a base in the flank or in the motif, and run it through thousands of different genomic contexts to see what the average effect is. From that, you can derive position weight matrices that look very comparable. Here, this is for the motif. You can see that it's extremely similar to what we observe in vitro, where it's just TF and DNA, but it's trained only on this KLF1 chip data. We can also do this for the flanks. And I was pretty astounded to see this because these are pretty subtle effects, but it definitely pulls out these proximal flanking Gs and the more AT-rich sequence in the flanks. And so our findings in vitro do seem to be relevant to how these TFs are binding within cells, but it's still a qualitative measurement, and we were curious, how much can these flanking sequences actually tune the fraction of molecules that are bound in cells? And to do that, we then actually went back to SMF, and so we built a library where we have 4 high affinity motifs. It has different flanks across the library, but along one molecule, each copy has the same motif and flanks. That's just so that we can get adequate signal within chromatin. We then install it at AVS1 and K562s. These are an erythroleukemia line, and so they have very high expression of KLF1. We then predict the energies of each of these sequences using our in vitro derived PWM model and feed it into these partition function models that I showed earlier in the talk. And so KLF1 also recruits BAF, and we can fit these parameters separately on a similar motif copy number style library. And so here, we're not training any new parameters to make these predictions. And what we predict is that across the library of different flanking sequences, we should be able to tune occupancy in cells from being completely unbound and nucleosome occupied to completely transcription factor bound. And indeed, that exactly matches what we see in SMF data. And not just by changing those proximal bases, but also we see this effect that as you have more GC content in the distal flanks, you get less occupancy. So in summary, for that second bit, we saw really big effects from sequence context outside of the motif. These in vitro derived preferences are really similar to what actually matters in cells. And we can deploy these thermodynamic models to be able to predict molecular states across not just motif copy number, but other aspects of motif grammars. And so thank you for your attention. And the most important slide is just acknowledging all of the people that contribute to this work, since these are really collaborative projects. So thank you, and I'm happy to take any questions.
Are those like repeated sequence motifs that you're kind of studying where there's kind of nice spacing in there, all motifs for are these synthetic circuits, or are they in regular circuits, or how like frequent is that, and do the motifs ever like change for one transcription factor than another, or like I guess, and how does, if so, how does like having multiple different transcription factors, how do you think about cooperativity in that sense?
Absolutely. That's a really good series of questions. So these are all synthetically designed sequences that we've installed. One caveat that was pretty subtle is that we use a GPC methyltransferase, so it only methylates Cs within a GPC context. And so the reason we get such highly resolved measurements is because we insert them in these synthetic sequences. There's now other approaches that go to deamination where you're not necessarily constrained to a GPC context, but just Cs, and that works probably better genome-wide. Most genomic enhancers are comprised of somewhere between 4 to 6 motifs is kind of the average range. And so it is reasonable in that range, but it tends to be that they're more diverse. So it's not necessarily one transcription factor at a time. It did necessitate like these more in-depth synthetic approaches to understand one transcription really well and understand both how the protein and its activation domains contribute separately to binding and potency. And so if we could characterize that more broadly across transcription factors, you could totally take these same kind of thermodynamic style approaches to be able to model that, but you do need a lot more measurements. Yeah.
So maybe extrapolating a little bit beyond what you've been able to do experimentally, thinking about how far the sequence context matters. Do you have a sense of, is there sort of a continuous decay of that, or is it actually the case that you can extend really far beyond these things and that sequence context still matters for expression?
So you do see that it matters less as you get farther away from the core motif. Something I didn't mention about that data is there's a really interesting periodicity in it. So in vitro, you see a correlation, an autocorrelation in the signal of affinity with the GC content that has a periodicity of 10.5 base pairs. And that's not because you're creating motifs in the flanks. And so the effects from that can go out 30, 40, 50 base pairs in the in vitro data. It becomes more subtle as you get farther out. But the way we've been thinking about it mechanistically is that there's a few models of how flanks can tune TF binding. One is you're like creating low affinity sites. We know in our data that we're not doing that. Another is that you're creating overlapping motifs with the core motif. So it's like high affinity and then some lower affinity ones overlapping. Our flanks also aren't driving that mechanism. There's also been studies that you are creating kind of repetitive elements where they're very, very low affinity, but so entropically favorable because there's so many of them. That also doesn't explain our data. And the periodicity in it is really pointing us towards a model where it's tuning TF search. And so part of how TFs explore DNA is through 1D diffusion. And so if that sequence context is tuning hopping parameters, that could explain why we see such distal effects up to 30, 40 base pairs away.
So one of the things that I love about the work that you presented, Julia, is how you're able to make these very simple fundamental models from rather sophisticated data. In some ways, that's a remarkable difference from all of the GPT type models where you take lots of data, come up with something that is fairly complex. Here you take lots of sophisticated measurements and you test those very fundamental, profound concepts with simple models. And I thought that's a wonderful thing for our community to see and think about how we can interpret our single cell protein measurements. And they do have a question, not just a comment. So my question is, when you think of the regulation of transcription and the type of models that you and your colleagues have made, do you think that quantification of the fraction of transcription factors bound to DNA versus not bound to the DNA in the nucleus can be helpful for constraining those models more and asking more questions?
That is an interesting question. So we do, like, clearly transcription factor concentration plays a really big role in the energetics of when you are bound or not bound. I do think it's an interesting question of how much time does a transcription factor spend unassociated to anything, weakly associated to cofactors in the cell, exploring DNA, or being specifically bound. And that super modulates the fraction of transcription factor that is free. And that is the parameter that matters for determining the thermodynamics of this. And so actually from the paired in vitro and in vivo measurements, where we're able to measure the in-cell effective concentration, which is that free fraction, and the in vitro energies, we can extrapolate how many molecules are free, which seems similar to the order of magnitude of which transcription factors are expressed. But it would be, I think, pretty interesting to know, actually, what percentage of your transcription factor is available for binding, because that is the information that would go into these energetic models.
Thank you.
Yeah, thank you so much.
This is sort of curiosity question. Do you see in your data cells or molecules that do funny stuff that don't really correlate with the linear relationship you saw? I wonder how that would come out in your data, but maybe you do some kind of filter for some noise or something like that, and maybe that correlates with a mutant or something. I'm just curious.
No, that's a great question. So we don't do any filtering. So that was all of the molecules that we sequenced that you saw in those plots. If you do a poor experiment, like if you don't lyse your cells completely and there's some cells that didn't get lysed, then you'll see that the methyltransferase didn't get in and those molecules are totally unmethylated. So occasionally if you didn't do an experiment well, you will see that. We don't throw those out even, we just tend to do a better experiment if that was the case. We have also done these experiments on plasmids, and then you start to see funnier things because plasmids are not nucleosomal the same way that you are when you're integrated within the genome. And so there things start to look a little bit funkier with a lot more spikes up and down and sparse nucleosomes. And so that is maybe more of like weird molecules that we see. But yeah, in general, from the data sets we have, it's pretty not filtered. Yeah.
Any additional questions for Julia? If not, let's give her a hand.
Thank you so much.